So those are the neurons, but what about nerves? Because we usually talk about the system as it relates to nerves. Now, what we have to remember here is that the nerve doesn't equal a neuron, and a neuron doesn't equal a nerve, even though we talk about it as such. When we talk about the nerve, we're talking about this structure here, in which we have a cluster of tissues. The cluster of tissues is surrounded by connective tissue, which is referred to as the epineurium. Inside the epineurium, we will see other clusters, which can be referred to as either fascicles of neurons or as a bundle of nerve fibers. These fascicles and bundles are surrounded by another set of connective tissue known as the perineurium. Inside the perineurium, we have sets of tissues which are encased within the endoneurium. The endoneurium is going to house what's referred to as the nerve fiber. The nerve fiber is the neuron. In the case of the efferent pathway, it's the axon. In case of the afferent pathway, it's the dendrite. Taking and transmitting that information to the spinal cord on the sensory side, or taking the information and transmitting it peripherally on the efferent or motor side. In order to get to where it needs to go to, these axons need to have a tube that stays open at all times. The tube that we will talk about when we talk about adaptations and repair and recovery is formed by the combination of the endoneurium and the Schwann cell. When we talk about the nerves, remember is that it's a composite tissue, so it's made up of neurons and neuroglia or glial cells. It's not one type of nerve, it's a whole different, it's not one type of neuron within the nerve, it's a whole bunch of types of neurons within the nerve. The neurons themselves will transmit via what's referred to as the nerve impulse. This nerve impulse will be transmitted at variable speeds based off of which type of neuron is being stimulated. Once that transmission hits the terminus, the passage will then take place via a chemical release at the synapse or the neurotransmitter. Each neuron within the nerve will transmit one type of neurotransmitter. The neuroglia within the nerve are going to perform a host of functions. The Schwann cells there will be involved with the myelination process. Other glial cells will be responsible for inflammatory responses and regulation of metabolism. The connective tissue that surrounds it, just like we saw with the image, is referred to as neuriums, where we have the outer being the epineurium, the centralized neurium surrounding the bundles and fascicles being the perineurium, and then the connective tissue surrounding the individual nerve fibers being the endoneurium. Now, one of the things that happens with axons is that they can integrate with some of the glial cells referred to as myelinating cells. And there are two types of myelinations that will take place. We have peripheral myelination by the Schwann cells, and we have central myelination by the oligodendrocytes. When we talk about the peripheral cells and we talk about the central cells, within that indication of myelination, we have what are referred to as myelinated and unmyelinated axons. The difference between the myelinated and the unmyelinated is whether or not the oligodendrocyte and the Schwann cell is able to encapsulate the axon with the membrane. All neurons will interact with a myelinating cell. Whether or not they are myelinated or not is dependent upon whether or not that myelinating cell is able to enwrap the axon with either projections or by the cell body itself. These myelinating cells ensure that the neural tubes peripherally stay intact and the organization of the axons centrally remains stable. So when we look at the peripheral nerves, we have distinct ways in which we look at the neurons. And so we have the myelinating neuron, where the Schwann cell starts to interact with the axon. As it interacts with the axon, it starts to walk itself around the axon. As it walks itself around the axon, it anchors itself. And it continues to walk and project itself. As it projects itself, the cytoplasm will start to be compressed within the enwrapped region where membrane and cytoskeletal proteins remain, but the bulk of the cytoplasm gets moved away from the encapsulating area around the axon. This usually happens with medium and large axons because there's enough membrane of the axon itself to allow for the myelin to wrap before it touches itself. The area of membrane of the Schwann cell that encapsulates the axon is referred to as the myelin sheath. The entirety of the Schwann cell itself will be referred to as the neural lemma. It will incorporate itself with the rest of the axon, sometimes referred to as the axonal neuronal lemma. Is basically the cell membranes for that part of the neuron. Now notice in the unmyelinated, the Schwann cell itself is unable to wrap itself around the axon. And that's because the axons are so small that as soon as the Schwann cell starts to wrap, it immediately touches itself. And that's the indication for the Schwann cell to stop projecting. On the unmyelinated axon, the Schwann cell itself will support multiple axons within the neural tube, whereas on the larger axons, we will see multiple Schwann cells projecting along the axon, and each Schwann cell will be wrapped multiple times around the axon. Now, something slightly happens centrally that we don't see, and that has to do with the space that's available. So peripherally, we'll see multiple myelinating cells per axon. Centrally, we see multiple axons per myelinating cell. And that has to deal with the fact that we don't have a lot of space, but we have a whole lot of cells. And so what the oligodendrocyte does is that it starts to project itself out just like what the Schwann cells did. But it projects itself out in multiple directions at the same time. And as it starts to encounter axons, it starts to wrap. And just like with the Schwann cell, if it's able to completely go around the axon before it touches itself again, it will continue to project. And as it continues to project, it will form the myelin sheath. These oligodendrocytes will support and connect with some of the protoplasmic astrocytes so as to stabilize the axonal projections within the central nervous system, particularly within the cerebral cortex, to ensure that the arrangement of the axonal projections remains stable and what's referred to as a radiated and radiating pattern. So what are some of the keys that we have to remember here when we talk about nerves and neurons? Well, neurons are aerobic tissues that are amitotic. We cannot dictate that a neuron is the same as a nerve, and we cannot dictate that a nerve is the same as a neuron. Remember, we're going to see multiple neurons within a nerve and multiple types of neurons within a nerve. Each neuron will be responsible for producing one type of neurotransmitter, and in some cases, it will only produce a single neurotransmitters. So when we look at the nerve itself can release a milieu, can release a whole bunch of neurotransmitters based off of which neuron within the nerve is being stimulated. Now, this stimulation and release is kind of key because once the neuron stops releasing any type of its neurotransmitters, it starts to undergo its apoptotic cycle. One of the reasons why neurodegenerative diseases occur is because of blockage on that release of the neurotransmitter, starting the apoptotic cycle for the neuron because it's unable to release the neurotransmitters. In terms of myelination and in terms of support of the axons, peripherally, we will be dealing with the Schwann cell, in which we'll have multiple Schwann cells per axon. Centrally, we'll have the oligodendrocytes, and with the oligodendrocytes, we're going to have one oligodendrocyte to multiple axons.